Hey guys, once again welcome to this channel HRS Saleem, and today we'll be studying about transport in plants. That is a new unit, plant physiology. And after a short break, I'm back to the form, and hereafter I will be assuring that there won't be any gap in between the videos. So every day you'll be getting the videos on time. Okay, and if you forget to subscribe, please do subscribe the channel and please share to your friends. So it will be helpful for the students who are preparing for Neet. Okay, so we'll start this chapter today, and in this lecture, okay, we'll be completing till short distance transport. Okay, we'll be completing till short distance transport, and the long distance transport will be followed in the next video. Okay, fine. So first we would like to know what is transportation in plant. Okay, first we'll have a small introduction. Okay, so usually many substances are being transported in flowering plants. Okay, let's say water. Okay, let's say water, mineral, organic nutrients, plant hormones. So all are getting transported day to day. Okay, and you can say hormones like growth hormones is all also getting transported, right? Okay, so this transport is usually classified based on a distance, how much it is getting traveled. Okay, if those substances are traveling for a long distance, okay, then it is called as long distance transport. Or if it travels for the short distance, then it is called as short distance transport. Okay, so you should, in a small way, in a keyword, you can say long distance means where the translocation will trace place. Okay, so translocation will takes place in the long distance, whereas in short distance you can say diffusion takes place. Okay, so diffusion is nothing but from higher concentration to lower concentration the substances will be moving. Okay, that is called a short distance. Okay, and when we talk about a direction of transport in root plants, when we talk about a direction of transport in rooted plants, usually they are of two types. One is either unidirectional or it is multidirectional. Unidirectional means only water. Okay, unidirectional means water and minerals. Okay, so which is present through xylem, which will be transported or supplied through xylem from root to stem. Okay, from root to stem the transport will be done, and that transport is unidirectional. That means from once, if the water moves from root to stem, then again it won't come back to the root. Okay, so if there is an excess of water, then that will be transpirated. Okay, that will be transpirated, right? Okay, so in any case that water will not come back to the root. That is called as unidirectional. For multidirectional, transport of organic and mineral nutrients. Okay, transport of organic and mineral nutrients, you can say food. Okay, mineral nutrients, usually they will be having a multidirectional flow. Okay, and here you can say this multidirectional will start from the sink to storage to sink. It will be keep on dividing, it will be keep on transferring one place to another place. Okay, so organic nutrients are usually synthesized. So this organic nutrients usually they are synthesized in leaves, and they are transported to all parts of the plants. Okay, and in if a part of plant undergoes senescence. What is senescence? There is one important word called as senescence. Senescence is nothing but aging. Okay, senescence is nothing but aging. Okay, so getting aged. Okay, so if a part of a plant undergoes senescence, then the mineral nutrients are relocated from that part of a plant to the growing part. Okay, that is multi-directional. Remember that the organic part, okay, organic part will be usually it will be produced in a leaves. It will be synthesized in a leaves. Okay, so once if that part undergoes senescence, if it is about to get aged or it is going to die, by the time the mineral nutrients will be relocated from that part to the growing part. Okay, so that is how the transportation starts working. Fine, so this is the basic introduction about the transportation in plants. Okay, we'll have a means of transport, that is types of transport. Okay, two types of transports are there. One is passive transport and another one is active transport. Passive transport and another one is active transport. Okay, so passive transport means where diffusion takes place. That means transfer of substances, transport of substances from one place to another through without any energy, then it is called as diffusion. Okay, so in diffusion what happens? See, transport of molecules or substances will move from high concentration to low concentration. transport of molecules or substances which move from high concentration to low concentration. So usually this passive transport is very slow process, and it also results in gas exchange. It also results in gas exchange. So this diffusion, usually this diffusions will be affected. Okay, there will be a concept like factors affecting. Factors will be affecting, like concentration gradient will leads to a lack of diffusion. Permeability of the membrane will leads to lack of diffusion. Temperature and pressure will also leads to lack of diffusion. So they are all the factors which affecting the diffusion. Okay, and the next one is facilitated diffusion. Facilitated diffusion is quite a bit complicated among the diffusion. Okay, it is also a type of diffusion, but what exactly is, usually we all know the membrane of the cells. The membranes of the cells are made up of lipid layers. Okay, the membrane of the cells are made up of lipid bilayers. So whatever that lipid soluble substances may easily move faster. Okay, it may easily move faster from the cell, from cell to outside the cell, or from outside to inside the cell. It will move faster. But some other substances which are hydrophilic moiety, that means they are less soluble. Okay, they are less soluble or insoluble. Okay, those type of substances can't move in this membrane. Okay, through this membrane. So by the time there will be a facilitated protein will present on the membrane. Okay, by the time a facilitated protein will be present over the membrane. Okay, and those membranes are called as membrane proteins, and they are also called as protein transporters. So those membranes, what it will do, it helps in transporting. Okay, it helps in transporting less soluble or insoluble substances. Okay, which will not work against concentration gradient. Okay, if it supports the gradient, okay, then it will move, but it will not work against the concentration gradient. So this is all an important point. Okay, so protein transports form protein channel. Okay, example, you can say porins. Okay, so what is porins? Porins are nothing but proteins. They are nothing but proteins which is forming a large size pores. Okay, which is forming a large size pores. Okay, so this porins will be present outer membrane of the plastids. Okay, it will be present in the outer membrane of the plastid. It will be present on the mitochondria and even on some bacteria, and which may allow even small side protein to pass through. Okay, that is all the role of porins. Okay, fine. And the next one is aquaporins. Okay, when we talk about water channels, there are such type of proteins called as aquaporins, which is all an integral membrane protein. They are all called as integral membrane protein. Okay, they are called an integral membrane protein, right? Okay, so when we talk about mechanism of action, what exactly this water channels or protein transporters or this diffusion, what exactly it is going to do? Okay, so that is called as mechanism of action. Very simple. Extracellular molecule which is attached to transport protein. First, the extracellular molecule, let's say this is the ECF. Okay, this is the ECF part. Okay, and this is the inner cellular part. Okay, inner cellular part. So here what happened, whatever the molecule attached to this, so let's say some molecules are there. Okay, some molecules are there. Okay, so those molecules will first come and attach to the transport protein. Okay, so what happened now, this protein will rotate. Now this protein will rotate. When it rotates, the molecule will move inside. Okay, and that will be released inside the cell. So this is what a basic idea about action, which is done with the help of passive transport. Okay, so here you can say three types of, based on a direction of transport, this passive transport is getting differed into three types. Okay, so one is you can say uniport. Okay, one is you can say uniport, where only one single type of molecule are transferred into the cell, where only one single type of molecule transported into the cell, that is called as uniport. Antiport, which is like if two types of molecules are transferred into opposite direction. Well, first we'll go to symport, it will be easy. First, uniport is one single molecule, one single type of molecule which is transferred into the cell. Symport means two different types of molecules are transported in the same direction, An antipode means two different type of cells, okay, two different types of molecules which are transferred into opposite direction. You can see here one molecule is entering inside, whereas another molecule is exiting outside. Okay, it is getting exited. Okay, so this is all called as passive transfer. So here what students wants to remember in Neet point of view, first they should remember what exactly diffusion is. Okay, diffusion is going to be a transport of molecules or substances. Okay, then what is the difference between facilitated diffusion? Facilitated diffusion is about lipid soluble particles. Lipid soluble particles will be easily diffused faster, whereas insoluble or less soluble hydrophilic moiety, okay, those will be they will transport through the membrane with the help of some membrane proteins, and they are all called as protein transporters. Okay, and they will be having a porins are nothing but the large size pores which is present over the membrane. Okay, and some aqua protein porins are also there. Okay, so this is what you have to remember for your NEET examination, right? Fine. Next we'll move to active transport. Okay, this is about active transport. So now in the earlier slide we saw what is passive transport. Here we'll be talking about active transport. Okay, so here during active transport, molecules are transported against concentration. Molecules are transported against the concentration gradient with a help of carrier protein, or you can say a membrane protein. Okay, that is an example. That is a difference between an active transport and passive transport. Okay, so passive transport will not work against concentration gradient, whereas active transport will work against the concentration gradient. Okay, so how it will work? It has pumps on the membrane. Okay, you can say it is a sodium potassium pump. Okay, so which will perform uphill movement. Okay, what is it uphill movement? Okay, it is very simple. They are the proteins. Okay, pumps are the proteins which participate in active transport of molecules. So the pumps will perform uphill transport of work against the concentration gradient. Okay, that is the role of the pumps. Okay, so you can say some common carrier proteins. Okay, we'll see some common carrier proteins. What is a similar character, or you can say what is a common character of a carrier proteins? Okay, so first they will be they participate in both active and inactive. They will participate in both active as well as passive. Okay, they will both participate in both active and transport. Okay, they are specific in transporting molecules. Okay, they are very specific in transporting molecules, okay, across the membrane. Okay, and the third one is these are sensitive to the inhibitors. Okay, these are sensitive to the inhibitors. Okay, that means like once the inhibitors are there, automatically the transport will be stopped. Okay, and they will be react with protein side chains. Okay, in all proteins, the last one is if all protein transporters are involved, okay, the transport rate reaches to the maximum value. Okay, it will reaches to the maximum value, and that is called as saturation. Okay, so this is one important question which been asked multiple times in NEET. Okay, what is that saturation? Okay, so how come the diffusion will keep happening? Okay, we will keep organizing the diffusion. Okay, the diffusion will organized in a form till it attains a saturated level. Okay, and after that it depends like whether reversible process will also be occurred. Okay, so in active transport, energy requirement is there. Okay, and energy is required for this transfer. Okay, energy is required in the transfer between the membranes. Okay, whereas in passive there is no energy requirement. Fine, so that is all about active transport. Okay, we'll have some small differentiation. What is common about simple facilitated diffusion as well as active diffusion? First, when we talk about energy requirement, yeah, diffusion does not require energy, whereas active transport requires energy. Okay, so next one is role of driving forces. Okay, so this diffusions usually they will not work against concentration gradient, so they will be working under a downhill movement, whereas ATP hydrolysis will undergo and it act against the concentration, and that is an uphill movement. Okay, and next one is membrane proteins are required. No, in simple diffusion there is no requirement of membrane protein, whereas in facilitated diffusion, yes, channel proteins are required, whereas in active also proteins are required. Okay, and next one, transport. Transport of saturation. Whether there is a transport of saturation, no. It won't happen. Why? Because diffusion is for small level distance, right? Okay, so like for few distance, for few amount, it will be keep on diffusing. Okay, but when you are talking about a facilitated diffusion, it reaches maximum. Okay, so this facilitated diffusion, it will be keep on progressing, and it will reach the maximum level. Until that, it will be keep happens. Okay, and active transport is also the same case. Okay, next one is selectivity. Okay, selectivity, whether it will be allowing all type of substances or it is selective for specific substances. Yes, see, diffusion is not specific for any substances. It will transfer any type of molecules, whether if any molecules are present in high concentration, that will move to the lower concentration. That is of simple diffusion. Whereas for facilitated diffusion, yes, they are specific. Even in active transportation, that is specific. Okay, so this is all what common factors about uh, short term. Okay, about active and passive diffusion, right? Okay, and did I missed anything? Yes, I missed one thing, which is inhibitor. Okay, so any effects will occurs due to inhibitors. Okay, yes, in diffusion there is no effect. There is no effect because of inhibitors, whereas here it will be inhibited. In both of the places. Here it will be inhibited, and that effect of inhibitors will react with protein side chains. It will be react with protein side chains. So this is all the common factors about active and passive diffusion, right? Next we'll talk about water importance. Okay, so why? Because now we are going to talk about potentiality of the water. Okay, let me give some important points of our water. Okay, so we all know cells are made up of cytoplasm, right? So cells is filled with cytoplasm, and the cytoplasm is dissolved and suspended molecules, which is actually water. Okay, the cytoplasm, which is actually water. Okay, so you can say in herbaceous plants, around 85 to 90% herbaceous plants are filled with water. Okay, even you can say some watermelon type of fruits which have 92% of water. So this water will provide a medium. Okay, it provides a medium. Okay, so that medium will helps to dissolve. Okay, that will provides a medium for dissolving. Okay, for dissolving the substances. Okay, so that is the second one. And here water absorption is also essential for absorption of minerals. Okay, if a plant need mineral, if a plant is in the need of mineral, then water absorption is mandatory. Water absorption is mandatory for mineral absorption too. Water absorption is mandatory for mineral absorption. Okay, and the next one is the content of water. The content of water in seeds. Content of water in seeds are less comparing to. They are less when it is compared to other parts of the plant. Okay, so content of the water will be very less in seeds and woody plants, whereas in succulent tissues, okay, that means plants which require lot of water, and those type of plants will be having a huge amount of, it will be having a water high content. Okay, so usually water becomes the limiting factor for growth. Okay, so this also helps as a limiting factor. Okay, for growth, limiting factor for growth. Okay, the growth attains only with the help of water availability, right? Next we are going to talk about water potential, solute potential, and pressure potential. So let me first explain what is water potential in a shorter way. Okay, let's say we have a pure water. Let's say we have a pure water. Okay, so this water, now there is only presence of water molecules. There is only presence of water molecules. Usually this water molecules will not be stable. Okay, it will be keep on moving from one place to another. Okay, the water molecules will be keep on moving from one place to another. Okay, so due to that movement, it will create an kinetic energy. It will create an kinetic energy. Okay, and it also, so this water potential will be is nothing but pure water will be having a lot of pure water will be releasing a lot of kinetic energy. So pure water is considered as having a highest water potential. Okay, make it very simple. Water potential versus kinetic energy, right? Water potential will releases kinetic energy. More water concentration, more kinetic energy. Okay, so pure water will be having the highest water potential, and is equal to, that is represented with 0. Okay, when we add any number of solutes, when we are adding a solutes on a pure water, so the water potential will be decreased. Okay, and this water potential is usually, it is expressed into a unit called as pascals. Okay, and that pascal is a unit which is which will be measuring the pressure. Okay, which helps in measuring pressure, right? Okay, this is all about water potential. Okay, when we talk about solute potential, it is nothing but a magnitude of lowering of water potential when the solute is added to the pure water. Okay, that means as we already said, in a water, in a pure water, when we are keep on adding some solutes, so what happened, the concentration of solutes will be increased. Okay, so higher concentration of solutes is lower the value of solute water potential. Okay, higher the concentration of solutes are is equal to lower the value of solute potential, so it becomes negative, and usually mostly it will be negative. Okay, always it will be negative. Make it very simple. Solute potential means magnitude of lowering of water potential when a solute is added to a pure water. Okay, higher concentration of solutes and the lower the value of solute potential. Okay, so it is negative. Okay, that is about solute potential. Next one is pressure potential. It is a mechanical pressure which is exerted, which is released out from the protoplasm. Protoplasm is nothing but cytoplasm of the plant cell. So due to the water entry, due to the entry of the water, let's say like a cell is there. Okay, so cell is filled with cytoplasm. Okay, a cell is filled with cytoplasm. Okay, so and let's say like the cell is kept under a solution. So over this like besides the cell, so water content is very high. Okay, let's say the water content is very high, so automatically what happens, the diffusion will take place, the water will enter. Okay, when the water enters the cell, okay, when the water enters the cell, the protoplasm exerts out from the cell. Okay, and which helps in maintaining turgidity, and when the water increases, pressure potential increases. When the water increases, pressure potential will also be increases, and usually it is positive in nature, right? Okay, so what is the relation between water potential, solute potential, and pressure potential? Water potential, to find a water potential of a water or a solution, okay, it is required to find the pressure potential. It is required to find pressure potential as well as solute pressure potential. Okay, so make it very simple. What is water potential? Water potential is nothing but pure water. Okay, only water molecules present in a solution. Okay, and that is called as water potential. It has a high potential when it creates high kinetic energy. Okay, when we are adding some solutes on that solution, when we are adding some solutes on that water solution, the purity of the water will be reduced. Okay, and it becomes negative, that is solute potential. Okay, when the water enters inside the cell, okay, and the pressure will be increased. Okay, pressure will be increased, and that is called as pressure potential. So to find water potential, we need solute potential as well as pressure potential. So that is what. Okay, in an atmospheric pressure, water potential is equal to solute potential. Okay, if the pressure is above atmospheric pressure, water potential is equal to solute potential plus pressure potential. Fine. Okay, so this is represented with a symbol called psi. Okay, so psi p, psi s, and psi wp. Okay, so that is how it is represented. Fine. Okay, next one, like we'll talk about osmosis. Okay, for demonstrating osmosis, there are multiple experiments, but in your NCRT, there is an experiment which is called as potato osmoscope. Okay, so there is an experiment which is called as potato osmoscope. Okay, so in this potato osmoscope, you can see, okay, like in the initial level, some colored water sugar solution is present. Okay, let's say this is a sugar solution, right? Sugar solution present inside a potato. Okay, and that level will be marked with a pin. Okay, that will be marked with a pin. When this sugar solution, when this potato kept inside a water beaker, okay, the beaker which contains water. Okay, so what happened when the high water content which is present outside the cell, that moves inside the cell. So what happened, you can see here in what potato atmosphoscope final setup, more than the earlier initial level. Now you could see the level of the water increased inside the potato. Okay, and that shows that osmosis is an diffusion. Osmosis or diffusion as the process where it will occurs from high concentration to low concentration. Okay, then you can ask me, what is osmosis? Usually in a plant cell, movement of any solute and the solute potential are determined by cell membrane, that is stonoplast. Okay, the cell membrane, or you can say a vacuole membrane, is called a stonoplast. Okay, cell wall will not restrict the movement of water and solutes because it is permeable for both. Okay, so this osmosis is the diffusion of water which across a differentiality or semi permeability. Okay, where this osmosis helps in if a solution with high chemical potential to a low potential. Okay, so from high chemical potential to low chemical potential, the osmosis will move. Okay, the solution which have lower solute potential, the solution which have lower solute potential, okay, that will also be moved to the higher solution. Okay, so it is reversible. Okay, it is reversible, I'm saying. Okay, and the next one, a solution with higher water potential will move to the lower water potential, and the lower concentration will move to higher concentration. So this is reversible. This is reversible, right? So osmosis is reversible based on the high to low concentration. Fine. So this is how the osmosis is explained through the osmoscope explanation. Fine. So when we talk about osmotic pressure, okay, when we talk about osmotic pressure in plants, we are supposed to study about three terms, which is plasmolysed cell, flaccid cell, and then another one thing, turgid cell. Okay, let's see first what is plasmolysis. Let's see what is first plasmolysis. Plasmolysis, it can be, it is depends on plasmolysis. It depends on the surrounding of the cell. Surrounding of the cell, maybe of following types. Okay, what are that? Let's say isotonic. Let's say I'm drawing a beaker here, three beakers. Okay, so I'm drawing a three beakers. So here inside a cell is there. Okay, so you can see the concentration of water, inside the cell and outside the cell both are equal. Okay, inside and outside the cell, the level is equal, and that is called as isotonic situation. That is called as isotonic situation. And the next one is hypotonic. Hypotonic, if the solution is more dilute than that of cytoplasm. Let's say like a cell is there. Okay, the cell is filled with. Let me go with highlighter. Yeah, let's say a cell is filled with a beaker. Okay, and this has a high water content. Okay, so what happened, the water content will move from high concentration to low concentration, and this condition is called as hypotonic condition. Okay, so what happened, by the time the cell will start bulging. Okay, that is called as hypotonic. And the last one is hypertonic. Hypertonic means where the cell has lot of water content, The cell contains lot of water content, whereas outside the water content is very less. So by the time what happens, the water will be moved from higher concentration to lower concentration. That means from cell to beaker. So later what happens, the cell will become shrunken. The cell will become shrunken, and that condition is called as plasmolized. The shrunken cell is called as plasmolized. Okay, so I hope you will understand what is plasmolized. Okay, when a water is released from the cell, when a water is released from a cell, okay, the cell will start shrinking, the cell membrane will start shrinking, and later the cell organelles will also shrink, and those type of cells are called as plasmolized. Okay, and this will done when it is in a hypertonic condition. Okay, and later when it undergo hypotonic condition, again it will be reversible. Okay, plasmolized cells are, again it is reversible. Okay, so the isotonic, when you talk about when again water enters inside the cell, again the cell will be bulged. Okay, and it will start creating a turgid pressure. Okay, and this is called as turgor pressure. Okay, so in this turgor pressure, what happened? It will maintain the cell shape. Okay, so usually plant cells will maintain the shell shape with the help of cell wall, whereas this turgor pressure will leads to bursting. It leads to bursting of the animal cell. Okay, so this turgor pressure leads to bursting of animal cell, whereas in plant cell the turgor pressure will not leads to bursting. Why? Because cell wall will be maintaining that rigidity. Fine. So that is all about osmosis. Okay, I hope you understood what is this. So I'm just making a note, just understand. Plasmolysis is nothing but a water movement, okay, out of the cell. Okay, from the cell it is moving out. Okay, when the cell is placed under a hypertonic condition, when a cell is placed under a hypertonic solution, as a result the plant cell and cell membrane shrinks, and the cell wall will also be, cell wall will remains. Okay, so it will shrink and it will go away from the cell wall. Okay, so during plasmolysis, first water will be lost, and then cytoplasm will be lost, and then after that vacuoles. Okay, so whereas when it is kept under hypotonic solution, again it will be reversible. Again it will be reversible, and it becomes rigid, and it creates turgor pressure. Okay, so that is about osmosis in plant cell. There is another one condition. There is another one condition which is called as imbibition. There is another one condition which is called as imbibition. Okay, so imbibition is nothing but, imbibition is very simple. It is also a special type of, diffusion. Okay, so in this diffusion, this occurs between solid particles. Okay, that means a water which move, which is absorbed, the water which is absorbed by a solid. Okay, water absorption, let me write here, water absorption by a solid particles. Okay, solid particles, or you can say colloid particles. Okay, solid particles and colloid particles which resting in enormous increase in volume, so the size of the solid will also be increased. Let's say mountains are there, not a mountain, let's say two rocks are there. Okay, where some space is there. Okay, so when water enters in, when water enters inside, enters in between the spaces, so what happened, the rocks itself will break. Okay, rocks itself will be broken, and that condition is called as imbibition, right? Okay, for imbibition two properties should be there. Okay, what are all the two properties? First one is affinity. Affinity is nothing but observing capacity. Okay, observing capacity. And next one is water potential gradient, should be present. Okay, and some examples of inhibitions are absorption of water by seeds, and use of swelling woods. Okay, you could have seen the wood will be swelled during the time of winter. Okay, that is all because of imbibition. So this is how the short level of water movement will take place inside our plants. Okay, in the next video we'll discuss about the long distance transport of the plants, where it is highly complicated and it involves with specialized cells also. Okay, and moreover the long distance transport is very slow comparing to the short distance transport. Short distance transport will move very fast and it will reach to the maximum very fast, maximum condition very fast. Okay, whereas in long distance it is very slow process, and we'll discuss all those things in the upcoming video. And don't forget to subscribe this channel. Okay, so what is that? Our motto, is just to think big, dream big, and to achieve big. Okay, so hope you all would have enjoyed this session. Okay, if you have any doubts, please do comment under the chat box, right? Okay, so with that, I'm winding up this session and I will be happy to continue with the next lecture, right? Okay, thank you guys, bye.